Quantitative data is numerical data about quantities which can be measured and written down with numbers. Once this numerical data is collected, the researcher uses what is collected or observed into their research. For quantitative data, the information you need is numerical data, which is analyzed using statistical methods. If there aren't any numbers involved within the research, it can't be quantitative research. There are four main ways of collecting quantitative data, which are field-based experiments, laboratory-based experiments, observations, and questionnaires. This type of data is usually recorded systematically, so it can be analyzed within a computer database. Also, it can be collected by simple graphs which show the correlations between two or more items. Lastly, you can use cluster analysis, which is useful for identifying relationships between groups where there is no clear hypothesis. You should collect quantitative data via probability sampling because this type of data is collected at random, and therefore this type of quantitative data collection stops out the possibility of sampling bias. Also, you need a large amount of participants for quantitative data because it is essential that your research results are representative, as it is critically important that when you conduct quantitative research, you have a large number of randomly selected participants in each group you survey. This is because research will then be more representative and generalizable, and therefore more reliable, which will make it a better research. Once you have collected the data, you can compare it with other researchers who have done similar studies to see if you have got similar results and whether they have found anything different to yours. Types of experiments. The two types of experiment I am studying are pre-experiment and quasi-experiment. Quasi-experiment is an experiment with a random task that ensures that both the experimental and control groups are corresponding. Pre-test is a circumstance that is observed at two time points, one before the research and one after the research. Changes in the outcome of the study are supposed to be the results of the research. Once the research has been completed, there are three essentials of true experimental research that can be measured, which are control, randomization, and manipulation. Firstly, the control is there to stop the effect of extraneous variables from influencing the outcome of the research. This guarantees that the outcome is caused by the manipulation of the independent variable. Therefore, it is crucial to keep all other variables constant. Randomization is a random sampling of respondents into groups. Participants should have the same being sampled into the research. This ensures that the outcome of the study is due to the manipulation of the independent variable. Manipulation is when the researcher will purposefully change the independent variable, which is the condition that will be applied to the experimental groups. Experimental method advantages and disadvantages. The advantages are that the researcher controls the pace and location and environment of the experiment. Therefore, they will be able to allow the research to be taken in place in a natural environment, which is more valid and more realistic for the respondents. Also, this research is representative, and therefore the results can be generalized to the population from which the sample was taken. Also, if the researcher is able to control the confounding variables, then we will be able to see the cause and effect between the independent and dependent variable. The disadvantages are that most of the volunteers are overrepresented by the USA, UK males and university students, so it questions the generalizability beyond these points. Also, participants know their participant, and this may bias reaction and change their behavior because they know they are being researched on, like the Hawthorne effect. Finally, there are some limitations regarding ethics and health and safety, which are all important aspects. Experiments and groups. Intervention experiment is should have a very clear testable hypothesis so that there is no objectivity and the researchers know exactly what the research is about. Also, you need to be able to operationalize all concepts so that it can be applied to independent variables, dependent variables or covariables so that they are put into use. Also, it is important to try and control all extraneous variables, even though they are undesirable variables and are very hard to control. Post-test and pre-test experiments. The post-test is randomly assigned into groups, which are an experimental group and a control group. Neither group is pre-tested before the application of the research, but once the research starts to go underway, it is applied to the experimental group, and the post-test is carried out on both groups to assess the effect of the manipulation. Pre-test is where the respondents are again randomly assigned to either the experimental or the control group, but instead both groups are pre-tested for the independent variable. The experimental group receive the treatment, and both groups are post-tested to examine the effects of manipulating the independent variable on the dependent variable. Experimental groups. The experimental groups are groups in an experiment that receive variables being tested. Only one variable can be tested at a time to make sure that the test reliable. The experimental group is compared to a control group because it doesn't receive the test variable. Control group. The control group is defined as the group in a study that does not receive treatment by the researcher and is then used as a target to measure how the other tested participants do, and is used in an experiment as a way to confirm that your research actually works. Designs. Matched pairs. Matched pairs is when there are two equal groups of participants and each participant is matched with a similar participant in the other group, e.g. their age, interests, IQ. Then both groups then take part in different situations of the independent variable. As with the independent groups, this allows researchers to control the participant variables that might otherwise affect the result. Independent groups include participants who are only given one condition of the independent variable. There can be several groups of participants, but each group only takes part in one condition of the independent variable and does not repeat anything. This means the dependent variable will not be influenced by the other effects. Crossover designs are observational studies as well as controlled experiments, and participants are usually exposed to more than one treatment, but the second condition could be affected by the first. Repeated measures uses the same subjects within each part of the research, including the control, and therefore the same participant takes part in each condition of the independent variable, and therefore each condition of the experimental includes the same group of participants. However, this means participant variables are decreased. Also, it saves time because all of the same participants are used at this all the time. Experiments. Laboratory experiments are experiments that find investigations and see if a hypothesis is scientifically tested, but in controlled environments called laboratories. The advantages with this are you're able to control all the variables, which makes the test more valid. Also, the equipment is usually top quality, and therefore you can get more accurate and precise results. Finally, it has higher internal validity, which allows us to have more confidence as a researcher. The disadvantages are that it doesn't reflect the natural performance setting. Also, it's very expensive equipment and you need training to use this equipment. Also, finally, there's a loss of ecological validity and realism to the world environment, which is less valid. Field experiments are done in the everyday environment of the participants, e.g. real life. The advantages with this is that it's cheaper and more accessible because it is in your natural environment like a field. This helps to support ecological validity for your research. Also involves collecting data in an environment that simulates the one that the sport analyzes being played, which will increase validity. The disadvantage is that there's less quality equipment outside. Also, the research may take more time than it might be done if it was done somewhere else. Also, it is difficult to control confounding variables, which means that there's lower internal validity and therefore less reliability. National notational analysis and observations is where the study of movements, patterns, strategy, and tactics in team sports. The advantages with this is that it can show the strengths and weaknesses of the respondents. Also, it can compare or show improvements of the respondents, and because it's in the form of video, you can annotate later instead of right there at the time. The disadvantages are that you need lots of training. Also, you may miss some behaviours on the video. Also, finally, you may need several observers watching it at the same time so that you don't miss anything else. Questionnaires are research instruments that consist of a series of questions for the purpose of gathering information from respondents.